Today we're going to be discussing blood. In this particular lecture, we're going to be discussing the general characteristics of blood and blood plasma and white blood cells. Blood is the life-giving fluid that runs through all of your arteries and veins and is pumped by your heart. The pH of blood is 7.35 to 7.45, and this is actually quite important. We're going to be spending an entire lecture down the line on just the concept of the blood pH and how it is regulated. So when we take blood, we take a vial of blood and spin it down. It spins down fairly quickly into three distinctive parts. At the top, we have a fluid that is called plasma. That takes up about 55% of your blood. In the center is less than 1%, a very small portion of your blood is going to be what we call the buffy coat. The buffy coat includes white blood cells and platelets. And lastly, at the very bottom of the tube is the stuff that gives it its red color. We have what we call the hematocrit, which is the red blood cell or erythrocyte portion of blood. And this is going to have all of your red blood cells, your erythrocytes. This is going to be about 45% of your blood. We're going to start off with the concept of blood plasma, and this works us back to the idea that we have a non-living fluid matrix called plasma in blood, which makes blood a connective tissue. Usually when we think of connective tissues, we're thinking about something solid, such as bones or cartilage, and these structures will have a solid matrix and then living cells that are kind of buried within this non-living matrix. The same is actually true of blood. It has living cells, such as white blood cells, that are buried within a non-living fluid matrix called plasma. Okay, so the blood plasma is thicker than water, but it is mostly comprised of water. It's about 90% water. And it's going to have, one of the primary components of blood plasma are ions. We've discussed, well, I'll just list the ones that we've discussed before. There are more than this, but these are biggies. Calcium, potassium, sodium, chloride ions. And then we have another ion that is called bicarbonate ion, which is sometimes called sodium bicarbonate, which would be NaHCO3, but we are just going to use HCO3 minus. We're going to come back to the concept of bicarbonate ion when we talk about blood pH and how that is established. So keep that one in mind. So these are the big ions. You'll notice they all have charges. One of the things to keep in mind with ions is that all of the fluids of your body are going to be reflective of the blood plasma in terms of ion concentration. In other words, if you have something like a hypernatremia, where you have high sodium concentration in the blood plasma, you are also going to have high sodium concentration in the interstitial fluid and in the lymph and possibly other fluids of your body as well. So the reflection of what happens in the blood plasma also is reflected in other fluids of the body in terms of ions. However, it might be helpful to differentiate the concept of ions from the concept of proteins. It turns out your blood plasma has a high concentration of proteins. It's quite important, actually, that your blood plasma has lots and lots of proteins. So the protein concentration is actually special to blood, and other fluids of the body do not tend to be highly concentrated with proteins. So the big protein that is found in blood, it's a good one to keep in mind, is albumin. In fact, about 60% of your blood proteins are going to be albumin. Albumin has no other role well, it may have other roles, but the main role of albumin is to actually maintain the protein concentration inside of the blood. That's about it. About 36% of your protein concentration of your blood is going to be globulins. The big one that we focus on is going to be antibodies. It's quite important. It's a Y-shaped protein, very important. Free-floating, Y-shaped protein, very numerous, that circulates through your blood. And lastly, we have two more proteins I want to mention because they'll be coming back to us in the next lecture. About 4% of your blood proteins are called fibrinogen, and about 1% of your blood proteins are called prothrombin. I wanted to cover the roots here. An inogen, which we'll be looking at two more of these coming up. An inogen is some kind of a protein that's in its inactive form. So fibrinogen is an inactive form of another very important protein called fibrin. And prothrombin, the root pro means before. So prothrombin is a protein that, again, is an inactive form of another very important protein called thrombin. So fibrinogen is an inactive form of fibrin. Prothrombin is an inactive form of thrombin. Keep those ones in mind as we go through, as we talk about blood coagulation in the next video. All of these proteins, albumin, the globulins, fibrinogen, and prothrombin, are created within the liver. Now we're going to put blood plasma behind us for a bit, and we're going to discuss our white blood cells, also known as our leukocytes. So we're going to start off with the concept of the white blood cell count. And before we actually get into the white blood cell count, we actually have to think about a complete blood count. So if we think about this in a general terms, and usually if you go in for an annual checkup, or even if you just go to a physician saying that you feel a little sick, oftentimes they will do a CBC, or a complete blood count. This is a good way to draw a little bit of blood out of your system. It's not going to hurt you usually, and you'll be able to actually count, literally count the number of things in your blood in order to determine whether perhaps you might have an issue with, I don't know, liver enzymes, or, kidney function, or your glucose levels. There's lots of different things you can test for just by looking at the blood. Every time I go for my annual checkup, I get 3 pages of information based on a few vials of blood that were drawn. So this is a CBC, or a complete blood count. Now, the normal count for white blood cells is between 4,800 and 10,800 cells per millimeter cubed, which is the same as a microliter. Now, this is a tiny, tiny, tiny amount of blood. And so the numbers in the thousands, it sounds like it's not a huge number, but it turns out, when you think about one tiny little drop of your blood, one little microliter of your blood, and extrapolate that out to the idea that you actually have, on average, about 5 liters of blood in your circulatory system at any given time, the numbers are staggering. So we concentrate on a very small drop of your blood and just count the number of white blood cells in that particular spot, and then we can extrapolate out from there and think about your total health. So your white blood cell count is about 4,800 to 10,800 cells per millimeter cubed. Now, the first term we're going to come across is a situation called leukocytosis. Leukocytosis is where you have between, say, 11,000 and 50,000 cells per millimeter cubed. This is an elevated white blood cell count. However, this is considered to be a healthy situation. It usually occurs when you are fighting something off. You've been infected with a virus or bacterium or something like that, and quite naturally your white blood cells will elevate their numbers in order to fight that off. And then you can actually do a differential white blood cell count from there and determine which white blood cells are actually elevated in number in order to focus in a little bit more on maybe a particular type of virus that you're fighting against, for example, or a particular pathogen that you're fighting off, for example. So leukocytosis is a completely normal and healthy response to some kind of a pathogenic infection. So it's an elevated white blood cell count, but it's completely normal and is actually a really good sign that your immune system is doing great. So this leads us to the idea of what white blood cells actually do. White blood cells have many, many different roles, but by and large, all of them are involved somehow in fighting off pathogens or external insults to the body. Let's continue on with some of our terminology here. When we think about white blood cells, there's 3 terms that we usually use to describe their functionality and their abilities. One of them is diapedesis, the ability to squeeze their way out of blood vessels. It turns out that, usually when we think about white blood cells, we're thinking about them being in the blood, but it turns out it's really important to also allow them to leave the blood and enter into the interstitial fluid, where infections oftentimes will be. They'll be living in the interstitial fluid, so it's important for them to be able to squeeze out of there. Diapedesis is kind of an unusual process because white blood cells are relatively large and they have to squeeze through these tiny little cracks called intercellular clefts in the walls of the capillaries, but they are able to do that. That's one of the things that they are capable of doing. That leads us to our next term, which is positive chemotaxis. Once the white blood cells have left the blood vessels and have entered into the interstitial fluid, they need to be able to find the pathogens that they're chasing after, and they find them through a chemical trail that is left behind by the bacterium or by the virus or whatever kind of a pathogen. And in this way, they're able to follow that trail through positive chemotaxis. And the general way that white blood cells move is called amoeboid motion. If you think of an amoeba sort of blebbing through, this little protist that kind of blebs through, and kind of like a jelly-like sort of a movement by, putting out pseudopods and then retracting the other side and kind of blebbing through space like a big ball of jelly. That's kind of how white blood cells move. They're much faster than amoebas are, but actually that's pretty much exactly how they move. So it's called amoeboid motion. So positive chemotaxis combined with amoeboid motion allows them to make their way through all the interstitial fluids of the body pretty much in order to find any kind of pathogens and attack them. So let's talk about why it is that the body would develop more white blood cells. This process is called leukopoiesis, the development of white blood cells. And we start off with a stem cell called the hemocytoblast, which looks somewhat like this. And the hemocytoblast will then be able to differentiate into different structures. Now, the hemocytoblasts live inside of the red bone marrow. And so the hemocytoblast is the stem cell that actually leads to the differentiation of red blood cells, white blood cells, and platelets. So all of the formed elements of the cells are formed as a result of the differentiation of the hemocytoblast, this pluripotent, this neutral cell that kind of starts off the whole thing. So the hemocytoblast is the starter. Now the question is, why would the hemocytoblast ever differentiate into any of these things? And the answer is that the body basically sends signals into the red bone marrow to say, well, in this instance, we need more macrophages. In this circumstance, we need more B lymphocytes, for example. And so there are interleukins, there are colony-stimulating factors, there are different chemicals that are sent by different structures of the body. And then the hemocytoblast will respond to those signals and will differentiate into all of the different white blood cells we're going to be discussing here. So we're going to be discussing basically 6 endpoints here. And we're not going to be focusing on all the intermediaries, but we are going to be focusing on some of the early stem cells. So hemocytoblasts will directly divide into 2 different types of cells, the early stem cells that will eventually differentiate into white blood cells. The first of them would be myeloid stem cells, and the second would be lymphoid stem cells. The myeloid stem cells will then further differentiate into myeloblasts and monoblasts. The myeloblasts will eventually become our granulocytes, and the monoblasts will eventually become monocytes. So let's go ahead and follow the myeloblasts. The first of the myeloblasts that we're going to follow is the most numerous of our white blood cells, which is the neutrophil. So the neutrophil has a particular look to it. It has, the way that we can recognize neutrophils is that they have lobed nuclei. So I'm using a purple marker here to indicate the nuclei in this case. They oftentimes appear bluish or purplish. I'm going to show 4 lobes inside of here, but it can be anywhere between 3 and 8 lobes inside of a neutrophil. They tend to kind of look dispersed like this. So this unusual looking nucleus is a good way to recognize the neutrophil. Now, the neutrophil, as I said, is the most numerous of the white blood cells. In fact, it takes up about 50 to 70% of your total white blood cell count. So remember that I said we can take that white blood cell count and then differentiate it further, and that's how we determine how many neutrophils there are versus other white blood cells. The size of the neutrophil is relatively substantial at 12 to 15 micrometers in diameter. That's a slightly larger than usual size cell in our body. Now, the latest that I've seen as far as the lifespan of a neutrophil is about 5 days. They have a hard life. The purpose of a neutrophil is literally to consume pathogens. This is what we call your typical phagocyte. They can consume entire structures, and therefore their job is a little bit harder and they don't tend to live very long. We'll be talking more about the neutrophils down the line. The next of the white blood cells we're going to be studying, the next one that's differentiated out of the myeloblast, is going to be the eosinophil. The eosinophil is distinguished by its nucleus that has 2 lobes sort of tied together, sort of looks like a horseshoe or like headphones. The eosinophil takes up about 2 to 4% of your white blood cell count. The size of the eosinophil is slightly smaller than the neutrophil. It's about 8 micrometers in diameter, which is relatively the size of a red blood cell, which we'll be discussing in the next lecture. And the survival of eosinophils tends to be about 2 to 5 days. Again, kind of a hard life. Eosinophils are specifically set up to consume parasitic worms. They cannot endocytose them. So the way that neutrophils work is they will consume things like bacteria and literally endocytose them and phagocytize them, bring them directly into their cytoplasm. Eosinophils are smaller and they're actually attacking much larger structures than themselves. And so what they do is they attack with numbers. Eosinophils can surround an entire worm and eat it away from the outside. The last of the cells derived from the myeloblast is going to be the basophil. The basophil is distinctive because it has extremely large granules. The size of the basophil is about the same as the neutrophil, but the granules themselves inside of here are quite large, on the order of about 1 micrometer each, which is about the size of a bacterium. And so inside of the basophils, we have huge granules. The nucleus has a distinctive shape, but you almost never see it actually because the basophil has huge granules. Now, by the way, neutrophils and eosinophils also have granules, but they don't tend to be really distinctive features. So all three of these, neutrophils, eosinophils, and basophils, are considered granulocytes or granule-filled cells. But the basophils themselves, the granules, are quite distinctive. Now, basophils tend to be present during certain inflammatory reactions and especially during allergic reactions. They tend to take up a really small percentage of your white blood cell count, about 0.5 to 1%. And as I said, they're about the same size as neutrophils, so 12 to 15 micrometers. And lastly, basophils will live just for a few days. Let's go ahead and take a look at the last of our early stem cell stages that are derived from the myelin stem cells, and these would be the monoblasts. Monoblasts will differentiate into several. Again, we're not looking at our intermediary stages as much. The monoblasts will eventually differentiate into monocytes. The monocytes have a nucleus that can be somewhat distinctive in that it might have a slightly kidney bean sort of a shape. The really distinguishing feature of monocyte, however, is its size. It tends to be quite large. The cell itself is quite large. The monocytes are 3 to 8% of your white blood cell count. The size is the largest of the white blood cells at 21 micrometers. And the life cycle is a little bit hard to derive exactly. If it remains in its monocyte stage, it only lives for a few days. But if it further differentiates into a macrophage, now macrophages are actually pretty well known. A macrophage can actually live for months or even years. And one of the reasons why the monocytes tend to only live for a short period of time is because they undergo apoptosis as sort of a programmed situation. You don't want monocytes sitting around for too long. Macrophages, however, can last for some time. They can actually last for years. Sometimes that can give you some trouble. So monocytes tend to be more inert or inactive, and the macrophages play a really, really big role in the inflammatory response and in the immune system in general. So we're definitely going to be coming back to the concept of macrophages. It might be helpful to remember that although monocytes tend to die off after a few days, macrophages, once they are activated, once they are further differentiated into macrophages, they can live for much longer, and they have very specific roles. Let's discuss the last of our white blood cells. If we take a look at our lymphoid stem cells, which again, remember that hemocytoblasts, our original blast cell, will differentiate into myeloid stem cells or lymphoid stem cells. They are then going to become some kind of white blood cell. The lymphoid stem cells are able to differentiate through several intermediaries into lymphocytes. Lymphocytes are pretty well known. Lymphocytes are B lymphocytes or T lymphocytes, and they are responsible for really specific things in the immune system, which we will be discussing down the line. They're oftentimes just called B cells and T cells. They're really well known. B cells basically are responsible for the secretion of antibodies, and T cells basically are responsible for destroying tissue in your body that is infected or that is cancerous. All right, so lymphocytes take up a good portion of your white blood cell count. Anywhere between 25 and 40%, holy cow, I said 25 to 40% of your white blood cells are going to be lymphocytes. So a substantial portion. This would be the second largest number. Keep in mind the largest number is neutrophils at 50 to 70%, and lymphocytes are your second largest at 25 to 40%. The size of the lymphocytes is not much larger than a red blood cell at 7 to 10 micrometers in diameter. They can get larger than that, but usually when we're thinking about what we're dealing with here, we're thinking about ones that are about 7 to 10 micrometers in diameter. Now, the life cycle of white blood cell of lymphocytes is almost impossible to specifically calculate because there's many different roles that are going on here. If they could be living for just a few days, or they could live for months, or if you're thinking about memory cells, such as memory B cells and memory T cells, these actually could technically live for your lifetime, and so they could be very long-lived. We're going to look at two different disorders. We're going to take a quick look at leukemia and a quick look at mononucleosis. Leukemia is your blood cancer. As is true of any cancer, it's an overgrowth of cells. Leukemia is usually an overgrowth of white blood cells, although it can present in different ways. Generally speaking, we're looking at a white blood cell count of over 100,000, for example. So we're going way beyond our leukocytosis, for example. So the first type of leukemia, which is quite serious, is considered acute leukemia, and we talk about this being a cancer of the early cell stages. And what I mean by that is that leukopoiesis, when we're developing our white blood cells, sometimes things can go wrong. It's a complicated process. And if something goes wrong at the early stem cell stages, such as the myeloid stem cells or the lymphoid stem cells, then you might have an issue where the body is really trying to compensate because none of those fully differentiated cells are present. And so the body starts to create more and more of these early stem cell stages, and you end up with huge numbers of white blood cells that are incorrect and aren't functioning correctly. So the acute leukemia would be quite serious. It's very fast acting. It could be a fatal situation. An example is acute myeloid leukemia, which only has about a 35% 5-year survival rate. Now, the other type of leukemia is considered chronic leukemia. This tends to be cloning that is occurring in those later cell stages. It's going to be slower to act. It's not going to be as serious. You do eventually need to get it treated. It will not recover on its own. However, people can live with chronic leukemia for some time, and in certain circumstances may not even undergo treatment. Your white blood cell counts will not get quite as large as in the acute levels, and so your body's energies are not being put into making hundreds and hundreds of thousands of white blood cells that are incorrect. An example of chronic leukemia would be CLL, which is chronic lymphocytic leukemia, which tends to affect people who are a little bit older. All right, so our second disorder that we're going to be discussing is mononucleosis. Mononucleosis is, or it's oftentimes called mono. It's oftentimes passed through saliva. It is rarely fatal, but it does make you feel very sick and with kind of flu-like symptoms that can literally last for months. And it tends to affect sort of youngish people, teenagers or people in their 20s. And it is caused by the Epstein-Barr virus, the EBV, which we will be discussing more down the line. And the reason why we discuss mononucleosis in terms of white blood cells is because generally speaking, it specifically infects a particular type of lymphocyte, in fact, the B lymphocytes, and sometimes the B lymphocytes tend to harbor it for a little while, causing us trouble down the line.